students, welcome to Sunil's tutorial. I'm Sunil Milwani and today we'll be doing this chapter called as Control and Coordination. Let's see the structure of the human eye. Right? Now, the human eye is an organ of sight which is housed in a deep depression in the skull. You have a depression in the skull called as the orbits. It is attached to the orbits with the help of 6 muscles that allow the eyeball to move. Right? Do we get this in here? So first of all, where is it located? The human eye. It is the organ. It is the organ of sight which is housed in deep depressions. Deep depression of skull, which is housed in deep depression of skull called orbits. So that's where the eye is located. It is located in the orbit and it is attached. It is attached to orbit by 6 muscles that allow the eyeball to move. That allow the eyeball to move. Right? Now, the walls of the eye are composed of 3 concentric layers. Right? You can see this in the diagram, guys. Can you just see the diagram? We have 3 different layers: the sclera, the choroid, and the retina. You have 3 layers which form the walls of the eyes. Right? So I can say that the walls of the eyes, walls of the eye. The wall of the eye is composed. The wall of the eye is composed of 3 concentric layers. Now, what are the 3 layers? The first layer is the sclera. Right? Now, is a touch non-elastic fibrous coat around the eyeball. That's the outermost covering of the eye. Right? The sclera, it is a touch non-elastic fibrous coat around the eyeball. It is a fibrous coat around the eyeball and it constitutes the posterior. It constitutes the posterior 5/6 portion of the eye. Right? So the outermost layer is called as the sclera. Right? Now, the transparent and the circular cornea lies in front of the sclera. You can see that in the diagram, guys. Can you please see that in the diagram? You have here's your cornea, right? The transparent and the circular cornea lies in front of the sclera and it forms the anterior 1/6 part of the eye. It lacks blood vessels. Sclera lacks blood vessels and is composed of special type of connective tissues. That's what sclera is made up of. It is covered externally by a thin transparent membrane called as conjunctiva. Right? the muscle fibers are inserted into sclera for eyeball movement. That's how you are able to see in various positions, right? Eyeball movement. And the third, fourth and the sixth cranial nerves control the eyeball movement to observe in different directions, right? Light passes and gets refracted through the curved cornea and the rays then begin to converge. So that's your entire sclera, fine? So the behind 5/6 part is your sclera. I can say that the transparent and circular cornea lies in front of sclera and forms anterior 1/6 of the eye. Forms anterior 1/6 of the eye. Sclera, it lacks blood vessels. There are no blood vessels in it. It lacks blood vessels. But is composed of special type of connective tissue. That's what it is made up of, right? Next, it is covered externally by a thin transparent membrane. It is covered externally by a thin transparent membrane. It is covered externally by a thin transparent membrane called conjunctiva. Right? The muscle fibers are inserted into sclera for eye movement. The straightened muscle fibers are inserted into sclera for eyeball movement. That's the reason why you have muscle fibers there. And the third, fourth and the sixth cranial nerves. And the third, fourth and sixth cranial nerves control the eyeball movement. Control the eyeball movement to observe in different directions. Right? Light passes and gets refracted through the curved cornea. Light passes and refracted through curved cornea, and rays begin to converge. Right? So that was my outermost membrane, sclera. Next, let's move on to the next membrane. The next membrane that you have is called as the the second membrane that you have is called as the choroid. Right? Now, what is the choroid? Right? The choroid lies inside the sclera. Now, what does it contain? It's the layer just beneath. It's the second layer of the eye. Now, it contains blood vessels for supplying food and oxygen to the eye. Right? It is deeply pigmented and is connected in front. This layer is connected in front to a thick structure called as the ciliary body. Here's your ciliary body. So it is connected to the ciliary body. It bears blood vessels and muscle fibers which run in circular direction. What do I mean by it? They run in circular direction, that is parallel to the outer edges of the lens. Right? Here it is. They run parallel to the outer edges of the lens. The thread-like suspension ligament arises from the ciliary body. Here you would have a thread-like suspension segment that would arise from the ciliary body, right, which gets attached to the equatorial edges of the biconvex lens. The lens is then attached to this layer, right. At the junction of the sclera and the cornea, a pigment, circular, opaque and vascular coat arises from the ciliary body, which is your iris. Right? So at the junction of the sclera and the cornea, you would have this opaque junction that would come up, that would be called as the iris. Fine? Which comes in front of the lens and leaving an aperture which is called as the pupil. Fine? The iris consists of two sets of muscles. This iris is made up of two sets of muscles. One is called as the sphincters and the others are called as the dilators. Right? The sphincters are arranged in rings and their contraction decreases the size of the pupil. Their contraction would decrease the size of the pupil. If the size of the pupil decreases, that means you would have less amount of light entering the eye. The dilators are arranged in a radial manner such that and they increase the size of the pupil by contraction and by allowing more light. So one of them would decrease the size of the pupil, the other would increase the size of the pupil. Right? The pigment of the iris imparts color to the eye. So that is your second layer of the eye. Right? Can you just write this down? It lies inside sclera. Right, that's the second layer. It contains sclera, did not contain blood vessels. It contains a network of blood vessels. It contains a network of blood vessels. It contains a network of blood vessels supplying food and oxygen to the eye. Food and oxygen to the eye. Right? Next, it is deeply pigmented body. It is deeply pigmented. It is deeply pigmented and is connected in front. Connected in front to a thick structure. Called ciliary body. To a thick structure called ciliary body. So the front portion of the forehead is your ciliary body. Fine. Next, it bears blood vessels and muscle fibers. It bears blood vessels and muscle fibers which run in a circular direction. In circular direction. Right? Circular direction, what do I mean by they run in circular direction? That is parallel to the outer edges of the lens. Parallel to the outer edge of lens. Fine. The thread-like suspension ligament, you would have, this is your thread-like suspension ligament, which arises from the ciliary body. The thread-like suspension pigment that arises from ciliary body, gets attached to the equatorial edge, of the biconvex lens. This is your thread-like suspension coming out, which gets attached to the biconvex lens. Here's your biconvex lens. Okay. Now, so you have the middle layer, that the front portion of which is the ciliary body, from where you have thread-like suspension coming out, to which the lens is attached. Next, I can say at the junction of sclera and cornea, a pigmented muscular opaque and vascular coat, arises from the ciliary body. What we are talking about is the iris. So, what is the iris? It is at the junction of the sclera and the cornea. This is your pigmented, muscular, opaque, vascular coat that comes in, right? Called iris. If you want, I will write this, called iris. That's your iris, right? Ciliary body. The iris comes in front of the lens. Leaving an aperture called pupil. Right? The iris contains two sets of smooth muscles. The first one being the sphincter, and the second being the dilators. These are the two muscles that the iris contains, right? Each of them has a specific function. The sphincters are arranged in rings. And their contraction decreases. The size of the pupil. So as a result of which you would have less light entering the eye, right? The dilators are arranged in a radial manner. The dilators are arranged in the radial manner, and they increase the size of the pupil on their contraction, and they increase the size of the pupil on their contraction. So the sphincters decrease the size of the pupil, whereas the dilators increase the size of the pupil, right? The pigments in the iris impart color to the eye. The pigments in the iris impart color to the eye, right? Now, let's move on to the third layer. The first was the sclera, the second is the choroid, the third is the retina. Right? Let's talk about the retina. The retina is a delicate layer that forms the posterior two-third part. It forms the posterior two-thirds part of the eyeball, and it is sensitive to light. Right? It bears light sensitive photoreceptor cells. The innermost layer is made up of light sensitive photoreceptor cells, which are called as rods and cones. Fine. Rods contain purple colored photosensitive pigment, which is called as rhodopsin, which are more responsive to dim light. That means they are more responsible, that they enable you to see in less intensity of light. Cones contain violet colored photosensitive pigment called as iodospin, which function only in bright light. So when you are in high intensity of light, it is the cones that enable you to see the image. When you are in dim light, it is the rods that enable you to see the image, right? Cones are of three types, that is short wavelength, medium wavelength and long wavelength. The short wavelength cones are called as the sensitive blue cones, the medium wavelength are the sensitive green cones, and the long wavelength are the sensitive red cones. Fine. So that's how you are able to, it's actually the retina that enables you to see the image. Once the image falls on the retina, it is the retina that will convert the image into a form so that the electrical impulse or the impulse reaches the brain. Fine, do you get this in here? So it converts the light image into an impulse that can be sent to the brain with the help of these two pigments, which are rods and cones. Fine, do we get this in here? Retina, it is very delicate layer. It is very delicate layer that forms the posterior, two-thirds of the eyeball. Right, so the posterior two-thirds is made up of this and is sensitive to light. Right? It bears light sensitive photoreceptor cells. It bears photoreceptor cells. It bears light sensitive photoreceptor cells called the rods and the cones. These are the two sensitive photoreceptor cells that retina is made up of, right? Rods contain purple colored colored rods contain purple colored photosensitive pigment. Photosensitive pigment called rhodopsin, right, which are responsive to dim light, which are responsive to dim light. Fine. Next, similarly, you have cones. So the rods which are sensitive to dim light. Then you have cones contain violet colored photosensitive pigment called iodopsin, which function in bright light only. So rods function in dim light, cones function in bright light only. Right? Cones are of three different types. Cones are of three different types. Right? First, you have the short wavelength, which are responsive to short wavelength, sensitive blue cones. These are your sensitive blue cones. They are responsive to short wavelength. Then you have medium wavelength, the ones which are responsible to medium wavelength, which is sensitive green cones. And the third is long wavelength, the ones which are responsive to long wavelength. These are your sensitive red cones. So those are the three layers of the eye, right? Next, to the intermediate layer containing the neurosensory bipolar neurons. These bipolar cells snaps with the retina, whose axons bundle together in the optic fibers. So all of this is going to bundle together into the optic fibers, so that the impulse that is created by the retina can be sent to the brain, right? And this is then sent to the brain with the help of your bipolar cells, right? Next, so that was the three layers. I can say next is the, after the three layers is the intermediate layer containing the, you have this intermediate layer, is the intermediate layer containing, here's your intermediate layer, guys, can you see this? This is your intermediate layer. Next is the intermediate layer containing short sensory bipolar neurons. Containing short sensory bipolar neurons. Right? What do these neurons do? These bipolar cells, snap with the retinal ganglion cells, with the cells. That means they receive the impulses from the retinal cells whose axons bundle together. Whose axons bundle together. Whose axons bundle together as optic nerves. So you would have the axons of the retina that would bundle together in the optic nerves, and the bipolar cells would receive, the short sensory bipolar neurons or the bipolar cells would receive those impulses from the axons of the retina through the snap-like motion or through the snaps, right? Now, you would have a very small area that lies on the retina just opposite to the cornea. Can you just see this? You have a very small area just opposite the cornea, opposite to the center of the cornea, which is called as the yellow spot. It bears a shallow depression called the fovea, right? It contains only cones and no rods and gives distinct interpretation of the image, right? So here you have this region which is exactly opposite to the cornea, and this region is called as the yellow spot, right? Ideally you would have a clear image when light falls on the yellow spot. Fine, do we get this thing here? So I can say that the next part of the eye is the small area. A small area lies on retina. A small area lies on retina just opposite to the center of the cornea. Opposite to the center of the cornea, called yellow spot. Right? It bears a shallow depression. It bears a shallow depression. It bears a shallow depression called as the fovea, right? It contains only cones. Contains only cones. The fovea contains only the cones and gives distinct interpretation of the image. And gives distinct interpretation of the image. Fine. Now, outside the fovea, processing of visual information can occur within the retina. The entire visual whatever is the visual information that falls on the retina is processed within the retina only. Then, exactly opposite to the pupil, you also have another lateral to the fovea. You have another place which is called as the blind spot, which does not contain any light sensitive cells. So if the image falls on the blind spot, you will not be able to see that image, right? So here you have perfectly proper image that would be formed, and a distinct image that would be formed, and at the blind spot you would have no image formed. If light falls on the yellow spot, you would have an exact distinct image formed, and if light falls on the blind spot, you would not have an image formed at all. You will not be able to see that object. Fine, do we get this thing here? So you can say that outside the fovea, processing of the visual information within the retina. Then you have the blind spot, that is lateral to fovea. Lateral to fovea, there is blind spot which does not contain light sensitive cells. Right? Now, the cavity of the eyeball is partitioned by lens, ciliary body, suspension ligaments into the anterior and the posterior compartments. Here, this part, this is my anterior chamber, this is my posterior chamber. Right? So, your ciliary bodies, suspension ligament and they divide the eyeball into two parts, the anterior compartment and the posterior compartment. Both these compartments contain certain aqueous substance which is called as the aqueous. This is your aqueous chamber and this is your vitreous chamber. They are filled with aqueous and vitreous humor, which is kind of a plasma kind of a substance, right? With the help of which refraction would take place and you would have an image that would be produced, right? So just let's write this down and we'll come to the working of the eye. The cavity of the eyeball is partitioned by the lens, ciliary body, and suspension ligaments. Into anterior and posterior compartments. Right? These are called as the aqueous and the vitreous chamber. These are called as the aqueous and vitreous chambers. Right? They are filled with aqueous and vitreous humor. They are filled with aqueous and vitreous humor, which help to refract and produce image on retina. That was the structure of the human eye, right? Let's see the working of the human eye. How does the working of the human eye take place? Now, basically, before we actually move into the details, you would have light falling onto the cornea, guys. You would have light falling onto the cornea that would fall onto the pupil. Depending upon the intensity of light, the iris would contract or expand so as to allow sufficient amount of light. Now, as the light passes on from the anterior chamber, due to the aqueous humor that is present in the anterior chamber, certain amount of refraction or some amount of refraction of light would take place. That refracted light would fall on the lens. Depending upon the focal length of the light, depending upon the distance from where the light is coming, the intensity of light would determine how much light would would pass through the lens, through the iris. The iris would expand or contract to control the intensity. Then how curved the lens is going to be would be determined by the suspension ligaments of the ciliary muscles. If the image is close by, then a large amount of refraction would be required, hence the lens would be more curved. If the light is coming from a distant place, then less amount of refraction would be required, so the suspension ligaments would pull the lens and the lens would be almost flat, right? So thus the refraction of light is further controlled by the lens. The refracted light then passes through the vitreous chamber. The vitreous humor again allows refraction of light to take place, and an image is then formed onto the yellow spot. That image is then processed by the retina and is then sent through the optic nerves to the brain. That's your basic principle of the working of the human eye. Right? Do we understand this? Light falls on the cornea, gets refracted in the anterior chamber, gets refracted through the lens, gets refracted through the posterior chamber, that's your vitreous chamber, and falls onto the yellow spot, gets processed by the retina on the yellow spot, and is then sent as an impulse to the brain. The working of the human eye. Let's just write this down, guys. Working of human eye. Right? Light rays from the object pass through the cornea and the lens. Light rays from the object pass through the cornea and the lens, right? These are then focused onto the retina of the eye. These are then focused onto the retina They are focused onto the retina of the eye. The rods and cones contain photopigments, which are rods and cones contain photopigments, which are conjugated proteins. Right? They are composed of these rods and cones photopigments. They are composed of opsin, which is nothing but it is a protein, and retinol, which is a vitamin A derivative. Vitamin A derivative. So the photopigments contain opsin and retinol. Right? Light induces dissociation of retinol from the opsin. Light induces dissociation of retinol from opsin. Right? They separate out, resulting in changes in the structure of the opsin, resulting in the changes in the structure of opsin right? As soon as light falls on the photopigments, you would have retinol that would separate out from the opsin. As a result, the structure of the opsin would change, right? This causes a change in the permeability of the retinal cells. This causes a change in the permeability of the retinal cells, right? As retinol separates out from the opsin, the permeability of the retinal cells changes, right? This generates an action potential which is then carried by the bipolar neurons. This generates an action potential. As a result of this, certain amount of potential or very small potential is generated, which is carried via the bipolar neuron, right? This is carried by the bipolar neuron, right? By the bipolar neuron and the ganglion cells, and further conducted to the optic nerves. Conducted to the optic nerves. So as soon as you have light falling on the retina, you would have retinol separating from the opsin. As a result, the permeability of opsin will change, the structure of opsin will change. That will bring about a change in the permeability of the opsin, which would generate a small potential, which would be then carried by the bipolar neuron and the ganglion cells into the optic nerve, right? Which would be carried to the optic nerve and then to the visual cortex of the brain. And to the visual cortex of the brain. That's how the message would be transmitted. The neural impulses are analyzed. The neural impulses are analyzed and the image formed on the retina is recognized. And the image formed on the retina is recognized. Now, based on the earlier memory and experience, neural impulses are analyzed and image formed on the retina is recognized. So that's how you would have the image that is formed on the retina that will be reproduced in the visual center of the brain. So do we understand the structure of the human eye and the working of the human eye? Right, we'll stop this here for the day. Thank you very much.